Hello everyone. Welcome to the new session of Animal Kingdom Part 2. So in this session we are going to study about some of the phylum, right? So the first phylum that comes for the classification of animal is the phylum Porifera. So in the previous session we have completed we have talked about the different types of characteristics on which the classification of the animals depends on, right? So there are the basis, that is the cellular grade of organization, or tissue grade of organization, or the organ grade of organization, right. And after that, then we have talked about the symmetry, right? Then we have seen there can be asymmetry. There is no symmetry in the organism, or they can have bilateral symmetry, and also they can have the radial type of symmetry. Now we have also seen some other characteristics, like they can be a coelomate, right? They can be coelomate. The organism that are having a true body cavity, they can be coelomate, but sometimes there is no body cavity present in the organism, that is acoelomate. And after that, we have seen one of the phylum, that is the Platyhelminthies, in which pseudocoelom is also there, right? Now, another characteristic that we have seen, that is the presence or absence of the notochord, right? Now, well, let us start some discuss in this particular session some of the phylum. So, first phylum you took upon here is the Porifera, right? So we can see here some of the examples of the Porifera. Like the first one is the Cycon. So this is the Cycon. This is a Euspongia, and this is called a Spongilla. So all these organisms are pore bearing organism. First thing, that they are pore bearing organism. Their body is having small pores all over their body surface, and that is why they are called as pore bearing organism. Okay, so these are the primitive multicellular animals and have the cellular level of organization. They are not a very advanced type of organism. They are primitive, but yes, they are multicellular animals. It means that their body their body is made up of many cells. Okay, but that all the cells are working individual. They are not organized in a form of tissue, and that is why they are called as that they are having the cellular level of organization. They are having cellular level of organization. Sponges have the water transport or the canal system. This is one of the very important characteristics of the phylum. Okay. Now, all the organisms constitutively are called as the sponges. They are called as the sponges, and they are having the water transport or the canal system. When you see any sponge, okay, like this, right? So they are having all over the surface, they are having small opening. Okay, small opening. These are called as ostia. These small openings are called as ostia. And here at the top you are having one more opening, that is called as osculum. Now if you see the internal structure, right, if I draw the internal structure of this, then they are like flask shaped, but inside they are having one cavity, and this cavity is called as spongo seal cavity. seal Cavity, spongiocele cavity. Okay, now this minute pores, if you see here, these are the minute pores, that is called as ostia, minute pores, right? And they are having a channel in between like this. Now, if you see the internal lining, then they are having a very special type of cell here. If you see one of the special cell, then it looks like this. Okay, this is the collar type of things. So this is even called as collared cells. This is one of the cells that is found only in this particular phylum. So sponges have the water transport and canal system. So here what you see, they are having the water transport or the canal system. Water comes from this ostia, move inside it and comes here. When this comes, water is present all over here, then this water is having some nutrients in it, some food material of this sponges, right? So this collar like cells, what they do, they can trap those food material. They can trap those food material. They can even take the oxygen that is dissolved in the water. Okay, they can take the oxygen that is dissolved in the water. So this is very, very important characteristic, and it is forming a canal like system that helps in the transportation of water from outside to inside, right? So the water enters through the minute pores that is called as ostia. So these are the minute pores that are called as ostia. Minute pores that are called as ostia. And in the body wall into the central cavity. Now this is called as the central cavity, and this is the body wall. This is the body wall, and this is the central cavity, or even you can say the spongiocele cavity, right? Now from where it goes out through the osculum. So water when get enter na inside this spongiocele cavity or the central cavity, what happen, all these collar cells can take the nutrients, they can take the oxygen, as well as they are removing all the waste that is produced inside their body in this water only in their central cavity. And here this opening at the this particular opening that is called as osculum, it release out. When water comes out from this, all the waste material also release out from here, right? So this pathway of water transport is helpful in food gathering. In this way, they can take the food from the water. They can also take the respiratory exchange. The gaseous exchange is also happening in this particular water movement only, right? And also removal of waste. Now you can understand how this water transport is important for this sponges, right? They, it is providing the food material, is it providing the respiratory gas exchange, it is also helping in the removal of waste from their body. So this sponges is having this canal system that is very, very important. Okay, now here comes the porifera. Okay, that is the same phylum. Coinocytes or the collar cells line the spongiocele and the canal. Digestion is intracellular. So I'm just drawing the one of the wall portion that is showing this coinocyte cells, right? So these coinocyte cells are arranged like this. Okay, now they are having the nucleus, and these are also showing here the arrangement of brush like border that is making the collar of these cells, and they are having one this flagella like this. Okay, so this is very, very important. This is the cytoplasm or the cellular part of this particular coinocytes. So this cell is very important of the poriferans, and this is the key, you can say one of the key importance feature of this phylum. So you can simply say the coinocytes or the collar cell, because this type of cells are found only in the case of porifera, right? So the name of this cell is coino coinocytes. Coinocytes, or commonly it is called as collar cells. Collar cells. Coinocytes or the collar cells. Now, whenever the food material comes here, any food material, okay, any food material comes here. This, with the help of flagella, they take this food material inside their collar and ultimately it reaches to the cellular part of the coinocytes. Right? So the coinocytes or the collar cell line the spongocoel cavity. So where is the spongocoel cavity? The cavity is like this. Okay, the cavity is like this. So I have just drawn a small part here. Okay, all over you can see the coinocytes are arranged inside like this. The inner wall lining is made up of this coinocytes or the collar cells, right? So digestion is intracellular, right? Digestion doesn't comes like take place outside. It is intracellular inside the cell. The body is supported by the skeleton made up of the spicules or the spongin fibers. Now, if you see the body wall, many spicules you can find like this. Okay, many types of spicules, or you can say the spongin fibers. These are also called as spongin fibers. So these spongin fibers are embedded in their body wall and they are like working as a providing it some sort of exoskeleton type of things, right? Because they are not having any skeleton inside their body, right? So the body is supported by skeleton, right? Made up of spicules and spongin fibers. Sexes are not separate, so they are hermaphrodite. It means that both male and female reproductive organ are present in the same individual, right? Both male and female reproductive organs are present in the same individual. It means that the eggs and the sperms are produced by the same individual, and the organism that are producing both the male gametes, both the type of male and female gametes by the same individual, they are called as hermaphrodite. So the sponges are hermaphrodite. Sponges reproduce asexually by fragmentation and sexually by the formation of gametes. So we see that in the case of sponges, sometimes what happened due to the water current, there is a wear and tear in the sponges, right? Sponges are like this. It is growing in a group. It is growing in a group. There is a rock and these sponges grow like this. Okay, now sometime what happens, some of the sponges get detached from it, it moved to the other places. Now they find a suitable substratum and they can get attached to it. So this is just the fragmentation. This is a asexual mode of reproduction. Now there is another mode of reproduction in the case of sponges, that they form gemmules. Gemmules. Okay, asexual reproduction by the gemmules also take place in the case of poriferans. Now, if you talk about the sexual reproduction, then for the sexual reproduction, they form the male and female gametes. Now, these gametes are released in the water, right? They are living in the water, so they released in the water, and fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult, right? So in fertilization is internal inside the body and the development is indirect. Indirect means that is a formation of larval stage. Formation of larval stage is seen here. So indirect, having a larval stage which is morphologically distinct from the adult, right? So adult, they are quite different from the adult. Now, if we talk about this examples of the poriferan, then we are having the cycon. This is the scientific name, that is the scypha, spongilla, right? That is also known as freshwater sponge, and euspongia, that is the bath sponge. Okay, so Sycon is the scientific name, Scypha, Spongilla, common name, Spongilla. This is the scientific name. Commonly, it is called as freshwater sponge, and use spongia, that is also called as bath sponge, because we use this sponge in a bathing. Now, another phylum we are having here is the Coelenterata. Coelenterata is also called as Cnidaria. This phylum is also known as Cnidaria. They are aquatic, mostly marine, and sessile or free living, radially symmetrical animal. So, if you see the symmetry of this particular animal, then you can see that this is a sea anemone, and this is the uh, Aurelia, Aurelia jellyfish. Now, if you see any of the jellyfish like this, okay, when you cut from here, you can cut it from here, divide into two equal halves. Again, you can cut it from the here, divide into two equal halves, right? So there are many, you can say, the line of division through which you can pass it to the center of the organism and divide it into two equal halves. Same in the case of sea anemone also. If you see the sea anemone, it is a cylindrical like this. It is having lots of antenna like this. Now, if you can divide from here, from the here, then you can divide from this side also, right? From this side also. So number of symmetry, because it is cylindrical now. So you can divide like this, 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 right? The center of symmetry is giving you a number of you can say this division of line through which you can divide the organism into two equal halves. So they are showing the radially symmetry, and they are sessile or free living. Sessile means they are attached to the substratum. So here you can see the sea anemone. It is attached to the substratum. Substratum. Sessile means attached to the substratum. And here you can see in the case of this Aurelia, it is moving in the water. So it is free living. Free living. So they are mostly marine. All the organism, mostly they are marine. And all are aquatic. There is no exception. They all are aquatic. And in the aquatic, mostly they are marine. The name Cnidaria is derived from the cnidoblast or cnidocytes, which contain the stinging capsules or the nematocyst present on the tentacles of the body. So let us understand what this. So this phylum is also having one this particular cell that is showing the characteristic of this phylum. Like in the previous one, the Porifera is having the collar cells or the coinocytes. That is the unique feature of that particular phylum. In the same way, the Cnidaria is having this cnidoblast cell. Cnidoblast cell. So let us see how this Cnidaria look like. So I'm just drawing you can say a hydra. Hydra belongs to this. So it is attached to the substratum. Now we are having the tentacles. Hydra is having tentacles like this. Right? This is the bud. Now, if you see this tentacle here, now this tentacle is having one very different cell. Very different cell like this, and it is having a stinging like thing, which when in a, you can say, resting condition, it is like this. But as this tentacle comes in contact with any, you can say, microorganisms or any food particles, right? What happened to this coinocyte cell? this coinocyte cells take out this stinging part outside, and it can take any organism. Okay, it can take its prey and move it inside the cell. So this is a nidoblast cell or nidocytes which contain the stinging capsule or the nematocyst. This is also called as nematocyst. So that is present on the tentacles and the body. It is also present on the body surface as well as on the tentacles. So this is very, very important, as this particular cell is helping into capturing the food material. It is helping in capturing the food material. And also it is keeping away from their you can say enemy. So that is mean that they are providing protection. It is also providing protection. Sometimes the organism can come and try to take this hydra to be eaten, right? To take its prey. Now, what they do with the help of this nematoblast or the nematocyst cells, they protect their body, they protect themselves. So, this is providing the protection as well. So here you can see the nidoblast are used for the anchorage, defense, and the capture of prey. So this, with the help of this, they can capture their prey. They can also help in the anchorage. Suppose the substratum is there, how to get attached to the substratum. Okay, they want to encourage at one particular substratum. So this is also helping in the anchorage to get hold at this particular surface. So with the help of this nidoblast cells, they can get attached to any surface. That is helping in the anchorage, and also it is helping themselves for defense mechanism. So here you can see this is the cell. It is having the cytoplasm. It is having a cavity inside which this needle-like structure is present. So when this needle comes out, it can take away its prey. It can helps them to get the defense from any other organism that is very harmful to them, or helps them to anchor to any surface also. Cnidarians exhibit the tissue level of organization and they are diploblastic. Now, the first time the tissue came in this particular phylum. So, in the previous phylum, there is no tissue-like things formed. Only what is there? The cellular things. Means in the cavity of the spongocoel, the cells are present. This was the thing which we have studied now. So this is made up of cells only. Okay, diploblastic. No mesoderm is there. Now, in the case of Cnidaria, one step is ahead. Now, they are having a tissue level of organization. Tissue organization you can see in the case of Cnidaria, but they are diploblastic. That is, they are having the ectoderm and endoderm layer, right? The outer one is the ectoderm and the inner one is the endoderm. So when they are in a developmental stages, these two layers can be seen. So that is why it is called as diploblastic. It is called as diploblastic. They have a central gastrovascular cavity, and with the single opening, the mouth or the hypostome. So if we draw any Cnidarian, so we can see the gastrovascular cavity like this. It is having a cavity, right? They are having the tentacles, and this is the mouth. So if they find any prey, this tentacle find with any prey, it can capture with the help of nidoblast. It can move its this tentacle and such that that this prey which was captured with the help of nidoblast get enter inside this gastrovascular cavity. Why it is called as the gastrovascular cavity? Because the digestion is taking place inside this cavity. And vascular means they are having the circulation of water inside it, right? So with a single opening, the mouth on the hypostome. So this particular region is called as hypostome. Hypostome. And in the hypostome, what is present there? The mouth. The mouth is present. Okay. Digestion is extracellular and intracellular. Both the type of digestion we can see. Means sometimes the digestive food material is given out and they take the digestion occur and then they absorb the food material. But sometimes they take the food inside it and the digestion take place in the cell. So this is a very, very important, right? Tissue level of organization. First time we see in the case of Nidaria, and they are also showing the diploblastic. Okay, diploblastic, it means that when the fertilization takes place, diploblastic means when the fertilization takes place, the zygote is formed. Now the zygote divide and redivide. Zygote divide and redivide and form a many number of cells. Now these cells arrange themselves into two layers: ectoderm, endoderm. Two layers, ectoderm and endoderm, and that makes the whole body of Cnidarians. And that is why the Cnidarians are called as diploblastic. Only two layers are formed at the time of development: ectoderm and endoderm, no mesoderm. Right? The central gastrovascular cavity, spongiocele cavity or the central cavity in the case of Porifera. Here, gastrovascular cavity is fine. This is different from the Poriferans, right? Some of the Cnidarians, here we can see the corals have a skeleton composed of the calcium carbonate. This is very, very important. Corals are also belongs to this Cnidaria. Okay, corals belongs to this Cnidaria, and their body is made up of they have a skeleton that is composed of the calcium carbonate. Whole body's deposition of this calcium carbonate. Cnidarians exhibit the two basic body forms that is called as polyp and medusa. So some of the organisms show only one type of this body forms, like in the case of Hydra. When you see Hydra, it look like this, right? So when the body is in the form of cylinder like this, okay, this is the polyp stage. The body form is polyp type. Now, there are some organisms that also show this medusa type. Medusa type, what they say? In medusa type, they are like in the umbrella shape like this, and they are move from one place to another. That is mean they are free living. Free living. And polyp is, you can say this is sessile. Okay. So this Adamsia is also showing this polyp stage. Okay, sea cucumber, that is showing the polyp stage, and this jellyfish Aurelia, that is showing the medusa stage. Right? So this is one of the vegetative phase, you can say, and this polyp medusa is showing the reproductive stage. Right? And one more thing, there are many organisms in which we can see both the stages. That means half of the life cycle is showing this polyp stage, wherein another half is showing the medusa stage. Right? So such things are seen in the case of Cnidaria. This is again one of the unique characteristics. The former is sessile. Okay. So this polyp is sessile and cylindrical, that we see in the case of Hydra, Adamsia, okay, whereas the latter is umbrella shaped and free swimming like Aurelia or the jellyfish. This we have already discussed, that the polyp, okay, the former means that is the polyp stage is the sessile attached to the substratum, and an example of such type is that is the Hydra and Adamsia. Sea cucumber, sea cucumber, whereas the latter umbrella shaped, okay, Aurelia is the umbrella shaped, that is the jellyfish. Now those cnidarians which exhibit in both the forms exhibit alternation of generation, that is the metagenesis. Okay, so cnidarians, just now I told you that in some of the organism they are having both the stages, both the forms you can see in one particular organism only. So that is the alternation of generation, that is the metagenesis, that is the polyps produce medusae asexually and medusae forms the polyp sexually. For example, obelia. Okay, very unique type of things. So do remember these things, polyp and medusa. Okay, Physalia, commonly it is called as Portuguese man of war. Adamsia, sea anemone. Penatula, sea pen. Gorgonia, sea fan. And Meandrina, that is brain coral. So these are very, very important. You must remember the scientific name as well as their common name, because sometime in the examination, as the following question can be asked, right? So here in one column one, they can give the scientific name, and in the column two, they can give the common name. Now you have to match. If you memorize this, then you remember this, then you can answer that. So this is very, very important. Next phylum is the Ptenophora. Okay, Ptenophora. So Ptenophores, commonly known as sea walnuts or the comb jellies. So they are also known as sea walnut or the comb jellies and are exclusively marine. Very important point. All the Ptenophoras are found in the marine environment only. They are again radially symmetrical, so you can see the walnuts are cylindrical like this, and you can have this comb plates. 1, 2, 3, 8 comb plates can be seen here, and along with this, they are having the this locomotory things. Now, this is also having the flagella attached to it. So this help this ball-like organism to be with the water, right? So this comb jellies are exclusively marine, radially symmetrical, diploblastic organism with the tissue level of organization. Again, the organism is diploblastic. It means that they are having ectoderm and endoderm layer at the time of embryo formation. No mesoderm is there, only ectoderm and endoderm. So, and that is why they are called as diploblastic organism. Diploblastic. Now they are also showing the tissue level of organization. So Porifera, they are having the cellular grade of organization. Cnidaria, tissue level of organization. And again, the Ptenophora is also showing the tissue level of organization. So these are the points that you must remember. And this is also showing the advancement of organism as they are moving from one phylum to the another. The body bears 8 external rows of the ciliated comb plates. So here the green in color, what you see, the green in color here, these are the comb plates. Okay, 8 ciliated comb plates are there. Comb plates. So in the examination, it can be asked, Ptenophora, comb plates, match the column in this way, right? So comb plates is a unique characteristic of this particular phylum, and that helps in the locomotion. Okay, so this is the example of Ptenophora, Ptenophora example is the pleurobrachia. So this is almost cylindrical, and they are showing the bioluminescence phenomena. They shine in the water. When they are present in the marine water, they shine. So digestion is both extracellular and intracellular. Okay, both the type of digestion is there. Sometimes they show the extracellular mode of digestion, and sometimes they show the intracellular mode. Bioluminescence, the property of living organism to emit light. They give light, and you can see them in the case in the dark, that these are the Ptenophoras. Okay, so they are well marked in the Ptenophores. Sexes are not separate, so they are again hermaphrodite. Both the both the sexes, that is the male and female, are present in the same individual. Reproduction takes place only by the sexual means. Again, a very unique feature. No asexual reproduction takes place in the case of Ptenophora. Reproduction takes place only by the sexual means. It means that they produces the male and female gamete that unite together from the zygote. Zygote arrange themselves in a two-layer diploblastic, and that originally forms the whole organism. Fertilization is external with the indirect development. Again, indirect development, it means they are having the larval stage. You have to remember all these points. These are the characteristics of this particular phylum. Now, what are the examples? Pleurobrachia and Ctenoplana are the example of this. Pleurobrachia and Ctenoplana. These are the example of the phylum Ptenophora. Now, next phylum is the Platyhelminthies. Platyhelminthies. Platyhelminthies, it means that flatworms. Flatworms. Okay, you can see the worms. The worms are there, but they are very flat. You can see that it is having the dorsal and the ventral surface, like leaf. The plant leaf you see now. Okay, dorsal surface, and the lower one is the ventral surface. Ventral surface. This is the dorsal. In the same case, here also, in the case of flatworms or the Platyhelminthes, they are having the dorsal surface and they are having the ventral surface, right? So they are, that is why it is written here, dorso-ventrally flattened body. Their body can be two surface. Upper one is the dorsal and lower one is the ventral surface, and hence that is why it is called as flatworms. They are known as flatworms. So here the two examples has been represented of the phylum Platyhelminthes, that is the tapeworm and the liver fluke. Okay, two examples are there, tapeworm and the liver fluke. So these are mostly endoparasite. Mostly they are endoparasite. They are found inside the body of the animals, including human beings. Including human beings. They are found inside the body. Flatworms are bilaterally symmetrical, triploblastic, and acoelobate animal with organ level of organization. Okay, so they are bilaterally symmetrical. You can divide the body into two equal halves, and that is why it is called as bilaterally symmetrical. Triploblastic. First time the organization of the embryo from the triploblastic layer. It means that body can be divided at the time of embryo formation. The cells arrange themselves into three layers of the cells: ectoderm, inside, endoderm, and middle one is the mesoderm. So first time we see the triploblastic layer in the organism, that is called as triploblastic. And acoelomate animal, no coelom, no body cavity is there, and with the organ level of organization. So first time we can see the organ level of organization. Again, these are some of the very unique feature: bilaterally symmetrical, Triploblastic. First time the organism is coming with the triploblastic layer. Acoelomate. No body cavity is found in the case of this animal, and that is acoelomate. Organ level of organization. Organ level of organization. This is again the very unique feature. First, before this, we have just seen the tissue level of organization. Now here the organs are formed. First time, hooks and suckers are present in the parasitic form. So in the case of in the parasitic form, they are having this hooks and suckers through which they get, with the help of hooks, they get attached to the tissue, and with the help of suckers, they absorbs the food material. They absorb the nutrients from the host. Some of them absorbs nutrient from the host directly through their body surface. Okay, they are absorbing the nutrient from their general body surface. But sometimes what they do, they get attached with the help of hook to the tissue, and with the help of suckers, they can suck the food material. Specialized cells called flame cells help in the osmoregulation and excretion. Now they are having one very unique cell. First time this type of cell is there. Flame cells. Okay, flame cells. So it helps in the osmoregulation. It maintains the balance of water in the body. And it is also helping the excretion, whatever the nitrogenous waste material is there, that is removed out from the body. So this is done by the flame cells. Sexes are not separate. They are not separate. It means that they are hermaphrodite. They are having the both the male and female sex organ in the same individual. Fertilization is internal. Okay, no external fertilization. Fertilization is taking inside their body only, and the development is through the many larval stage. This is again a unique feature of this phylum. Many larval stage. Still here, we have seen that every phylum is having at least one larval stage. In the case of Porifera, Nidaria, okay, we have seen in the Ptenophora also the development is indirect. Indirect means they are having one larval stage, but here they are having many larval stage. Okay, so some members like planaria possesses the regeneration capacity. If you take the planaria and cut it into 4-5 pieces, each piece will give rise a new individual, right? So it is having the regeneration capacity. Now, if we talk about the examples, then we are having the tinea, that is commonly known as tapeworm, fasciola, that is the liver fluke. Okay. So this is all about this session. Thank you very much.